I'm Yokogawa from Kuraray, Japan-based chemical company. I would like to show you multiple spheroid analysis method for cardiotoxicity testing with human iPS-derived cardiac cells under a micropatterned Elplasia 3D cell culture plate. This table shows several methods for cardiotoxicity testing. Ion channel profiling technologies include binding assays, flux assays, fluorescent membrane potential assays, automated and conventional electrophysiology. Further assay development in combination with human IPS technology have positive impact in cardiotoxicity area. For further analysis of this table, we have created a graph in order to better interpret and visualize the table. This graph interpretation, the x-axis represents the throughput and the y-axis represents the level of information content from low to high. We understand that you seek even greater throughput and even higher information content. There is a need. There is a challenge and opportunity in order to improve technologies. For example, the need to increase throughput, information content, and cost effectiveness. Let us share with you a Elplasia new microwave design approach in order to help your research. We propose multiple spheroid assays for cardiotoxicity testing. With typical culture method for cardiomyocyte, one data set is produced from one spheroid in one well. With a Elplasia plate, you can get multiple data points from one well. Now imagine each data point is a beating heart. You might be asking yourself how we did it. We utilized these three key elements of cardiotoxicity testing, namely Elplasia 3D cell culture plate, imaging instrument of Yokogawa, and human iPS cardiac cells. Let us share with you some novel aspects of Elplasia. Kuraray's 3D cell culture plate is branded and called Elplasia. Elplasia has many microspaces on the bottom of the surface. Elplasia's pattern is 200 micrometer wide and 100 micrometer deep. Please let me share with you a movie about Elplasia. Cells are gathering in each space. Cells form spheroids within 24 hours. 200 spheroids can be cultured in one well of 384-well plate. As you can see, multiple spheroids equal multiple data points, thus very high throughput and high content. This shows Yokogawa's confocal imaging system. It is called the cell Voyager. The features of this high-end model are the following: low photo bleaching, low phototoxicity, and wide view sensor mirror. Of course, Yokogawa's confocal system is real-time imaging with fluorescent. By using both our Elplasia plate and Yokogawa's machines, along with CDI's IPS cardiomyocyte, we have developed a new method for cardiotoxicity testing. This slide shows the methodology. The cells were prepared with materials and method of CDI. 60,000 cells are seeded per well of 96-well plate on non-adherent surface grade SQ type plate. For the test, The early cardiotoxicity kit from molecular devices was used. It made direct measurement of intracellular fluorescent change due to changes in intracellular calcium concentrations. After 7 days culture, the images were captured by cell Voyager at 1 hour after the compound were added. E4031, verapamil, propranolol, isoproterenol, sotalol were used for the test. The compounds were diluted to this concentration. To evaluate the beat per minute and early after depolarization of spheroids, the fluorescent intensity were observed by 20 frames per second for 30 seconds with cell Voyager. This is a figure before the compound were added. The cells formed spheroids like this figure. You can see small dot. This is a spheroid. The diameter of each spheroid is about 100 micrometer. In the 96 wave plate, you can make 700 spheroids in each wave. The fluorescence signal was recorded for 30 seconds. This video shows that cellular relaxation occurs independently. The cell Voyager and Elplasia SQ type enable us to observe a lot of spheroids under a wide-view sensor mirror. These videos show the changes of beat per minute with several compounds. In the figure 2, isoproterenol and figure 3, verapamil, which are known to increase the contraction rate, show increased beating rate compared to control. On the other hand, propranolol, sotalol, and E4031, which are known to decrease the contraction rate, show decreased BPM. Fluorescence signal in each spheroid was also captured by cell Voyager. Like this figure, this chart shows beating pattern of one single spheroid. In this case, the data of 16 spheroids were taken. In many ways, you can imagine these are 16 beating hearts. This is the data output from the Yokogawa instrument. In this case, the upper figure in each column, please note that a single green dot or a single blue dot represent a single spheroid. Green color dots represent 5 to 20 BPM, and the blue color dot greater than 20 BPM. Also, the number of average is O dot. Isoproterenol increases the heartbeat and shows a BPM of 42.79. Verapamil, which increases the heartbeat to an average BPM of 23.14. Propranolol decreases the beat to an average of 15.93 BPM. Sotalol, which also lower beat to an average of 14.40 BPM. E4031 also lower beat to an average of 16.36 BPM. The pie graph shows percentage. As you can see, I divided the BPM into two sections. In the control, 25 of spheroids beat at greater than 20 BPM, and 75% of spheroids beat between 5 and 20 BPM. In figure 2 and 3, the blue area percentage, which represent higher than 20 BPM, is larger than the control. On the other hand, figure 4, 5, and 6, the green area which shows between 5 and 20 bpm, increased compared to the control. This shows the frequency of occurring early after depolarization. Each chart represents a single spheroid. Please note, some spheroids do not show EADs. We produce the same conditions, however, some spheroids show different function. As a result, we calculated 82% of spheroids EAD is detected. As for the rest 80% of spheroids, EAD cannot be detected. As the data shows, Elplasia enables us to analyze frequency. That will help us an objective analysis. Thank you for your attention.